and we'll finish up chapter 37. So we looked at metabolism of fatty acids and a little bit of synthesis. What about regulation? So you can imagine that regulation of fatty acids, anything, might be important for our population with a huge amount of obesity. So melanoCoA is the precursor for fatty acid synthesis. Without it, you don't have lipogenesis. And it also inhibits fatty acid oxidation. That makes sense, right? If you're going to synthesize fatty acids, you're not going to want to oxidize them at the same time. And so it really does make perfect sense. So melanoCoA is synthesized by this acetyl-CoA carboxylase, also known as ACC. So there's two isozymes. Liver and adipose tissue have isozyme 1. Cardiac and skeletal muscle have isozyme 2. And the 2 seems to be quite heavily regulated. It's activated by high glucose and insulin. So does that make sense? High glucose, you got to take it out of the blood. You got to put it somewhere. Maybe it's a good time to store energy as fat. Insulin also does that. Insulin takes glucose and triglycerides out of the bloodstream. Glucose, of course, turns into starch, glycogen. Insulin causes triglycerides to be stored as fat. So anyway, there's three important effects related to this enzyme. So activation is going to increase melanoCoA. That's going to promote fatty acid synthesis and inhibit fatty acid oxidation. Inhibition of this enzyme decreases melanoCoA and thus inhibits fatty acid synthesis and promotes fatty acid oxidation. And I put this in red because this enzyme is inhibited by exercise. We all know the importance of exercise on losing weight. This is one of the key enzymes here. So how is it inhibited by exercise? Well, it's quite complex, but exercise activates an AMP-dependent kinase that then phosphorylates it, reducing levels of melanoCoA. So we had an interruption there, didn't we? First time that's happened. So where were we interrupted? Was it before this? I was talking about.
Last thing I heard was, yeah, you put that in red.
Okay. So anyway, mice whose gene for this enzyme was knocked out using RNA interference technology could not get obese no matter what you gave them to eat. So this enzyme responds to levels of insulin, glucagon, and glucose. So in the fed state, which means that you're not starving, high insulin promotes fatty acid synthesis. In the fasting state, high glucagon activates lipolysis, and adipocytes promoting fatty acid oxidation. So here's the reaction. Here's the enzyme. It's activated by insulin and glucose, inactivated or inhibited by glucagon and exercise. So if you're going forward with this because of high insulin or high glucose, you'll have a high concentration of melanocoenzyme A. That's going to promote fatty acid synthesis, inhibit fatty acid oxidation. If you inhibit that enzyme, by glucagon or exercise, you're going to have a low concentration of melanoCoA. That's going to activate fatty acid oxidation. In other words, you're going to burn your fats, and it's going to inhibit fatty acid synthesis. It also turns on carnitine palmitoyltransferase. Well, what does that do? Carnitine, if you remember, was the amino acid that's used to activate fatty acids for fatty acid oxidation. Brings them into the mitochondria. So anyway, this is something that's probably going to be a lot of research done on in the near future. Probably a lot of it's going on right now. So what about dieting and caloric restriction? So now we're getting a little bit away from the biochemistry, getting more into the physiology. But of course, they're obviously related. The department that I'm in right now is the Department of Anatomy, Biochemistry, and Physiology. So how do we lose weight, and what do you mean by losing weight? Usually we mean losing fat deposits from adipose tissue. Nobody wants to lose muscle mass. And so if you go to the gym and you're just working out, sitting there talking to your friends and stuff like that, it's unlikely that you're ever going to lose any weight. Especially if what I've seen when I rarely go to a gym, but I did go there one time for a meeting. And I walk in and I notice that, oh my God, everybody is just dressed so perfectly. There's a total fashion statement to be made when you go somewhere to try to work out and sweat. And I'm watching these people on the treadmills, and they're sitting there talking to each other, and then they drink their little Starbucks and everything. And I'm like, yes, you're actually undoing what you're trying to do. When you exercise, you're trying to burn you're trying to burn fatty acids, right? Triacylglycerols. You're trying to burn glucose. So here's the problem. If you go in there and you're trying to lose weight and you start drinking anything with sugars in it, you're going to have a high spike of glucose in your blood, which is going to cause the release of insulin. What does that do? Well, here's glucose in your blood. Here's your muscle and liver cells. High glucose in your blood does what? Causes the release of insulin. So insulin puts glucose back into the cells. Glucagon and adrenaline take it out, right? So they're counteractive. But what people don't also know is that insulin takes triglycerides in the blood and puts it back into fat cells. So you do not want the insulin effect when you're trying to lose weight. You want the glucagon or adrenaline effect when you're trying to lose weight and exercise. Do most people ever get to the point where they're actually burning fats? I would say probably not. I used to surf with a guy who was a physiologist. He had a master's in biochemistry. And we'd go out surfing on a kind of a bigger day on the North Shore. And you're simply paddling the whole time you're out because the currents are so strong. And anyway, so we would kind of like be out for an hour and a half, 2 hours of intense exercise. You're huffing and puffing. And then finally we would get like to the point where we're feeling not very much energy. If we stayed out for another half an hour, we start to regain energy. And that's when we thought we were starting to be able to burn triglycerides. But that's like an hour and a half, 2 hours of intense exercise before you ever get to that point. This is actually supposed to be chapter 39 now. So there is evidence, if you look at different organisms, caloric restriction makes organisms live longer. Okay? So we'll talk about that a little bit later. But yeah, species that have low metabolic rates consume less lipids, have less acidic ketogenic metabolites in their bodies, and tend to live longer. It correlates with different species, but there's no evidence that caloric restriction in humans will extend life. Probably a good diet will, and that's where we'll go in the next chapter, or in chapter 39, actually. Yeah. So any questions about anything in this chapter?
Did we talk about when your glycogen storage is full? Like what happens to your body once that mechanism of making more glycogen is full?
Well, if you keep bringing in calories and you can't make any more glycogen, then it's going to be stored in other ways, such as fat. And that is the problem with most people nowadays. Make sense?
Yeah, it makes sense. I just didn't know if we talked about like a cycle that it goes into.
Well, it's not a specific cycle. It's just basically one thing gets turned off and the other thing gets turned on. So the whole series of cycles are either down-regulated or up-regulated. Come on, open up. Here we go. Step. So this chapter is this never going to be complete because there's so many diseases and there's so many things that are constantly being discovered about diseases. But I wanted to give you an idea of what you might be able to think about doing your extra credit papers on. And I want you to see all the stuff that we've learned, how it can be put into use in solving diseases. So some diseases are called metabolic diseases. So there's just something wrong with the metabolism. Many of these are called genetic diseases. So you take diabetes, for example. Is there a genetic propensity for people to get diabetes? Yes. It's not a very clear one. So oftentimes metabolic diseases are a combination of genetic issues and environmental issues. Environmental means, you know, what you eat, how you exercise, not just what air you breathe. And then we have several storage diseases. Where there's a metabolic problem results from the accumulation of some biochemical. And that biochemical that we're talking about could be a fat-soluble vitamin, in fact. That will also cause problems. So then we get to chapter 39 on health and nutrition. That's kind of like the opposite. So something can be detrimental, that would be a disease. Not enough of it could be a problem with, you know, a needed nutrient. And so sometimes a disease comes from the effort to repair or protect the organism. It doesn't function right, such as an overactive immune system resulting in an autoimmune disease. So what about diabetes? So I took it into several different parts so we can look at different parts of it. So pancreatic alpha cells release glucagon. Pancreatic beta cells release insulin. And so we know that. Okay? It should probably be, I better check that chapter. So what is type 1 diabetes? Well, it's insulin dependent. So the beta cells do not release enough insulin, so the patient has to inject insulin to counteract the effect of glucagon. So the alpha pancreatic cells are not attacked and not destroyed anywhere near as often as the beta ones. Okay? So this disease is not linked to diet. It has both genetic and environmental components, obviously. But here's the problem. Too little insulin causes hyperglycemia. Too much causes hypoglycemia. So, okay, type 2 diabetes is not insulin dependent. Cells become insulin resistant to inadequate insulin receptor proteins. So that may not necessarily simply be the gene that codes for the receptor protein. It could be any of the other, machinery that's involved in, the production of insulin receptor proteins. So let's look at a few figures. So here's a glucose tolerance test. Okay. So here's what a non-diabetic will do. So this is hours after you drink, a thick glucose drink, right? Glucose goes directly from your intestines into the bloodstream, and then they see it. So there's a spike that happens fairly rapidly, and then the non-diabetic will be able to remove that glucose from the blood, right? And then there's a slight rebound over here. But what about the diabetic? It simply shoots up very, very high, and then it takes a long time for recovery to get back to normal. So normal is somewhere over here. Okay. So let's see. So here's another little diagram here. So let's see, here's the blood. So here's glucose going to muscle. Here's glucose or glucagon going to the liver. So here's the pancreas, right? So here it's releasing insulin, it's releasing glucagon, and the glucagon stimulates, wait for that to go away, glucose synthesis and export. Insulin stimulates glucose uptake and consumption. So here is the interesting effect. So here's normal fasting level of glucose in your blood, right? Somewhere between here and there. So hypoglycemia causes hyperinsulinism, fasting, starvation, whatever. So here you have, homeostasis over here. And then hyperglycemia is anything over this. So that causes fatty acid, cholesterol, body fats, and all this stuff to be made. And of course, these, we'll talk about, is basically ketosis. But here's the renal threshold. What that means is that this is when it starts showing up in the kidneys. In the urine. Okay. So you have this whole range of hyperglycemia that would not even be detected in the urine. So this is the reason why blood sugar has to be monitored. You can't use the urine. You're too late. Here's some of the effects of acute diabetic reaction. Excuse me. So lack of insulin or of insulin action, lack of insulin action. So regardless, reduced intake of glucose from the bloodstream, you get hyperglycemia. What is that called? Glucoseuria, glucose in the urine. Increase in production, excretion of nitrogenous wastes, all that kind of stuff causes dehydration. Okay? Increased catabolism of fats, hyperlipidemia, that means lipid in the blood. Then as it goes into what's called ketosis, where these catabolic acids and ketones and stuff like that get too much, it's called ketosis. And people know that if you smell acetone on somebody's breath, that's representing ketosis because that's one of the ketone bodies. And you get ketones in your urine, that also leads to dehydration. We get acidosis, which leads to dehydration. You start breathing rapidly, and basically you could go into a coma and simply die from it. Okay, so here's a couple of the things that's going on here. So the high catabolism of glucose produces lots of acidic metabolites, right? There's where the acidity comes from. Those are especially damaging to glucose permeable tissues, such as in the retina, kidneys, and the nerves. Then blood vessel proteins can become glycated. And so this is also a problem. And so we know that. And so you probably have seen on TV advertisements for different drugs and treatments where they're going to lower your A1C. What the heck is A1C? That's hemoglobin that has been glycated because of too much sugars in your blood. So glycation is known to damage beta pancreatic cells that are then responsible for producing the insulin, leading into a vicious cycle. So here's a very simple way to look at glycation. It's simply Le Chatelier's principle. So here's your reducing sugar, which is your glucose. Remember, it's a reducing sugar, so a reactive sugar. Here's protein. If you increase the amount of reducing sugar, you're going to force the equilibrium this way to get more glycated proteins, including hemoglobin. So, questions so far? So when you lower your A1C, what they're saying is that you're lowering this value because you're keeping this value low. So part 2, I put as ketoacidosis. And I see this right here has been moved. There's a problem all the time. It doesn't want to move in this mode. Anyway, so what is ketosis? So ketosis results from an overproduction of acetyl coenzyme A. So here's acetyl coenzyme A. And if this increases, here it goes. If I increase this, by Le Chatelier's principle again, I push the reaction this way. I will form more what's called acetoacetate. So 2 of these guys get together and make one of these, and it removes the coenzyme A. Well, so why is this a problem? Because this has no acidic hydrogen. After you take off the coenzyme A's using water, you produce the acidic forms of this. They ionize, and this is what causes acidosis. So ketoacidosis goes hand in hand because you're producing acidic intermediates from an intermediate that wasn't acidic before. Oh, another problem over here I see. Oh well, So I might be able to fix that. So here's acetoacetate with its acidic hydrogen producing acidosis. It can be hydrolyzed into acetone, and there's hydrogen still. And this acetone can be smelled in the breath. It's an indicator of ketoacidosis. It can also be reduced into beta hydroxybutyrate, which is also an acid, also contributing to acidosis. So, I mean, you know, your body can handle this stuff if it's not overly done. Acetone is removed from the breath. These acids right here can be metabolized. Any acidity can be alleviated by acidifying the urine, by adjusting your breathing rate. Let's see. So newly formed anions will require a counter ion, such as sodium. So the loss of sodium from the blood may throw off electrolyte balance due to hyponatremia. That's going to reduce the ability of the blood to protect against buffers. In addition, energy depletion itself contributes to acidosis. You take ATP and you hydrolyze it, yeah, you're putting more phosphate into the solution, and it contributes to acidosis. So if you go back and look at that last diagram you looked at, resulting in death, what's happening is that in clinical medicine, they're saying that the patient is crashing. That means that when one system gets out of whack, it shuts other systems down. So it eventually will lead to shock and then to death. You start messing around with vomiting and being dehydrated, you're throwing off electrolyte values, and we know that's very dangerous. So how do you prevent ketoacidosis? We have to teach the patient to predict a bad day, get them to realize what their body is trying to tell them, and that's hard to do. One of my oldest son's friends was diabetic. I watched him go through his teenage years trying to rebel against everything, and it took him quite a while to really settle down and learn how to moderate things.
How long can you be in ketosis before going into ketoacidosis? Is that very different for everybody?
Yeah, I mean, it's different for everybody, sure. Yeah, some people, yeah, I've noticed that some people just end up in the hospital. They don't pay attention. Yeah, the ambulance is picking them up off the street and they're in the hospital. Other ones are like, oh, I better go lay down for a while and drink a lot of water and everything. Make sure I'm hydrated and everything. Yeah. But yeah, it's something that has to be managed, you know?
My father-in-law is, he's diabetic type 2, and so he's done the ketogenic diet too. And so I feel like there's a fine line in between what's safe and what's dangerous.
And it's working for him, or?
It seems to be, yeah. He's lost a lot of weight and he's doing well.
Yeah, exercise is important. But what about some genetic diseases? Yeah, thinking about that, I have relatives in Waimanalo. They're my age, and they're in terrible shape. And it's really sad and makes me mad, but it's like it's all the diseases that are associated with, the Hawaiian ethnicity. But you know, I keep telling them, you guys have to exercise, you guys have to do this and that. And they're just like, they won't change their habits. They won't exercise. They think, oh, we're old now. It's like, no, you're not. But it simply is just a refusal to change their lifestyle. So I'm glad your relative is. Let's look at some things that are truly genetic diseases. So here's a hereditary transthyretin amyloidosis. It's a rare disease. A rare disease means that it affects only 50,000 people worldwide. So there's a mutated gene in the liver that causes a protein to be misshaped, and then it builds up in the patient's nerves and other tissues and organs. It forms amyloids, okay? So there was a drug approved in 2018, and it's an RNA interference drug. So it was the first RNA interference drug ever approved. It binds to the messenger RNA from the mutated genes, preventing the messenger RNA from producing the misshapen protein. The trick was that they made it into a double-stranded form of RNA to prevent it from instantly being destroyed by RNase. Here's another one. Pyruvate kinase deficiency. This is the last enzyme in glycolysis where PEP is converted into pyruvate. So it's a major control point for glycolysis. So inadequate production of this enzyme can be a major problem. And the disease is known as autosomal recessive. That means that the patient has to have 2 copies of the defective gene in order to have a true manifestation of the disease. Okay? So here's a carrier, or the father is a carrier, the mother is not. So there's one, let's see, I forget how it is. Oh, here it is, unaffected and affected. Okay, so here's one bad gene in one strand of the chromosome in the father. Then the mother has none. So the possibilities for your offspring is that you could have an effective, oh, this is dominant, I'm sorry. I should have put this first. So this is dominant if you only need one copy. So if that's the case, you got 1 out of 4 that could be given to your offspring, and you only need 1. So this person would be affected, that person will also be affected. But if it's recessive, like pyruvate kinase, then you end up with 2 carriers, one with not carrying it, and one's going to have both copies and would have the disease. So here's another one. So this one is not necessarily an inherited disease. This is what we call a chance mutation. 1 in 20 million people get this chance mutation. It's not inherited. And the mutation occurs in a gene that regulates the production of a protein that helps shape and support the nucleus and all the cells. And they start overexpressing a protein. So there is a drug that got approved in 2018. It's helping these people. Muscular dystrophy is a wasting of muscle tissue. There's a genetic defect in the gene for the protein called dystrophin. So it's found on the X chromosome. There's a frameshift mutation. A lot of these people die in their early 30s because of heart failure. There's other ones. Anyway, so they have tried to fix this in models in mice using CRISPR technology. They were able to do a few things. I didn't talk about that. Anyway, so dystrophin is a protein that's associated with the Z lines, and what it does is it kind of acts as a shock absorber in muscle contraction and relaxation. We did talk about it in the chapter on proteins. So I'm just trying to give you a quick survey of these things, and maybe you'll find something that sparks your interest and you want to research it. So accumulation of various metabolites. Well, one is lactic acid. You guys know that. You've experienced it. It's not detrimental. We have a way to get rid of it. But it can cause serious damage. So if you're a deep sea fisherman, you may know about this. So when you catch certain fish, they never stop fighting. Some fish kind of like, oh my God, I'm caught, I'm gonna die. They just kind of don't know what's going on, so they kind of go into shock and they don't really fight that much. Others fight until the very end. Tuna never stops swimming, even when you get them on the boat. And marlin, that everybody sees a lot about, you see these marlin jumping out of the water and they're trying to throw the hook and everything. But when they pull that marlin to the boat, they're able to grab it by the bill and unhook it and let it go. Why are they able to do that? And then they kind of hold on for a while, making sure water is going through its gill and hoping that it's still going to survive. Because that fish was fighting to the death. If you catch a marlin and that marlin dives deep, almost guaranteed by the time you get that marlin to the surface, it's dead already. It died because of not ketoacidosis, but respiratory acidosis and lactic acid. Lactic acid is an acid. It's stronger than acetic acid. And fishermen should know that when they pull a fish on board, if it's been fighting a lot and you're going to keep that fish, you should bleed it out, get all the blood out of it. Because all the lactic acid in the blood will go into the meat and actually acidify the meat and destroy the meat, quality of the meat. So, gout and kidney stones. This is an overaccumulation of different things. So much of, what's the word for it? Can't even think of the disease now. Okay, oh yeah, gout, okay. So gout is actually caused by uric acid buildup. And if you look at all these different things right here, adenine and guanine, those are the two purine bases that you find in RNA and DNA. So if you eat too much foods that have RNA and DNA, in other words, organisms like shrimp and maybe mushrooms and things like this that are rapidly growing and rapidly metabolizing, there's going to be a lot of adenine and guanine in the food. So that turns into xanthine and hypoxanthine on its way to becoming uric acid. And high levels of uric acid simply make these needle-like structures and start crystallizing out of your body fluids and causes severe pain. One of the treatments for this is allopurinol. So if you look at allopurinol, it's very similar to uric acid or to xanthine. I wish I had a picture that looked made it look a little bit better. But essentially what they did right here is change these two atoms around. The nitrogen, wait, no, not there, where is it? Oh yeah, right here. The nitrogen and the carbon right there are switched. Okay. So it becomes a competitive inhibitor to xanthine and hypoxanthine. So it slows the production of uric acid. So your body has more time to get rid of the uric acid and you won't possibly have the symptoms. So oxalic acid or oxalate is also responsible for some kidney stones. And there's a lot of oxalate in taro, some of the fruits that we eat in Hawaii, like star fruit, right? So spinach, rhubarb, beets, okra chard, sweet potatoes, yeah. So that can also be a problem. So how do you avoid having gout and kidney stones? Well, you remain hydrated. Okay, so even when you're working out and everything and you're sweating, you got to drink water all the time. Let's see. So phenylalanine and phenylketonuria, or PKU. So phenylalanine is converted into phenylpyruvate that then accumulates to in the cells. It's an alpha keto acid. It can acidify the cell. It's a competitive inhibitor to transamination. So it inhibits the conversion of amino acids into pyruvate, and it can cause all kinds of problems, especially if you are newborn. Newborns are screened for this disease by testing the urine and blood for the presence of phenylpyruvate. So it can cause mental retardation and all kinds of problems. And some people, even as adults, are sensitive to the artificial sweetener aspartame, because aspartame is a diamino acid. It's got phenylalanine, and it's got, what's the other one? Phenylalanine and aspartic acid. So when it gets hydrolyzed, the phenylalanine can be converted into phenylpyruvate and cause the problem. And then we have several lysosomal storage diseases. So there's about 50 inherited metabolic disorders from malfunction of the lysosome. It's usually a deficiency in a single enzyme required for the metabolism of lipids, glycoproteins, or mucopolysaccharides. Anyway, and then these substances that aren't taken up and pushed through the rest of the cycles build up. And this is what causes the problems. Yeah. So there's really no cures for this. It's just too complicated for modern day science. So here's a few other type of storage diseases: Tay-Sachs disease, gangliosidosis. What's that mean? Gangliosides, which are sphingolipids, accumulate in the brain's nerve cells. Premature death of the nerve cells. Niemann-Pick disease. There's several types. Buildup of sphingomyelin. Okay. This one is accumulation of cholesterol and glycolipids in the lysosomes. All kinds of very serious problems. Then very long chain fatty acids. Certain disorders of the peroxisome are associated with the accumulation of very long chain fatty acids. These are fatty acids whose chains are greater than 22 carbons long. They're too long to be catabolized in the mitochondria, so they have to be first oxidized in the peroxisomes. Okay? So anyway, so this takes quite a while to build up in the body, but eventually it does, and it leads to early childhood being disrupted by all kinds of different problems, and then eventually the people die. Maybe you heard of the movie Lorenzo's Oil. So that was some parents that had a kid with one of these disorders and had a buildup of very long chain fatty acids. And so these doctors made Lorenzo oil, which was a mixture of unsaturated fatty acids that competitively inhibits the enzymes and the formation of endogenous very long chain fatty acids. The efficacy of that oil was never proven. However, that guy lived 20 years longer. The kid lived 20 years longer than the average age. Anyway. So cystic fibrosis is an autosomal recessive disorder carried by mutations in both copies of the gene. It's for a membrane transport protein, okay, also known as an ATP binding cassette protein. It's a channel for chloride. And it's required for the production of sweat, digestive fluids, and mucus. And when that's not functional, secretions that are normally thin become instead thickened. So you have a buildup of all this mucus, and that's the problem. Okay. There's no cure. So when it starts affecting the lungs, that can be a problem. So the life expectancy between 37 and 50, but you basically die from lung problems. So you can imagine somebody with cystic fibrosis that were trying to live a normal life and they get hit by another thing that attacks the lungs like COVID-19, it could be the end. Cysteine and cystinuria. So here's cysteine. This is the normal amino acids. It can be, this is two of them, right? So that can be oxidized into what's called cysteine, which is the disulfide. So there's a transporter that can become mutated. Then it cannot reabsorb these amino acids, and so it starts to become concentrated in the urine, causing all kinds of kidney stones and everything else. That's why it's called cystinuria. So where would that come from? High concentrations of cysteine. Okay. So if you are suffering from this disease, what you should do is eat foods low in cysteine and drink plenty of water to try to wash this out. Then we have homocysteine and homocysteinuria, which means it finds its way into the urine. So, let's see. It's a homolog of cysteine, right? They're differing by one additional methylene group. Okay. So here is cysteine, here is homocysteine, here is methionine. So if you have deficiencies in the B vitamins, it can lead to elevated levels of homocysteine. So how would you treat this? Well, you would use your B vitamins. And here's another one that is also known to, betaine has been known to reduce homocysteine. So this is a major risk factor for cardiovascular diseases. So we know that homocysteine injures cells in the arterial walls. It's an inherited disorder in the metabolism of methionine. And so if you got homocysteine, you can convert it back to cysteine, or you can convert it into methionine. Okay, so you can see that just simply puts a methyl group on it, right? Yeah. So, oftentimes to assess your risk for heart disease, they will actually measure the homocysteine levels in your blood. And how would they reduce that? Well, they would give you B vitamins. But does that necessarily mean that you're now going to have reduced risk for cardiovascular disease. You see what I'm getting at? So cause and effect is not extremely clear in these cases. BMAA, I worked on this for a little while. One of our funding institutions, when I was working on chronic fatigue syndrome, got this idea. He read about the people in Guam, and he said, oh, well, maybe it's caused by this amino acid. So BMAA is beta-methylaminoalanine. So this is an amino acid, alanine, that's been modified. You don't find it in proteins. And anyway, this was causing all kinds of problems with the people of Guam. So it was a rather interesting story how they figured this out. So the older Chamorro people of Guam had maybe 100 times more neurodegenerative diseases compared to the rest of the populations of the world. And it was found that they made soup from the flying fox, which is a type of bat. And they also made flour from the seeds of the cycad plant. So both of those had significant amounts of BMAA. And BMAA is also produced in cyanobacteria. So that's also one of the problems when cyanobacteria get into the population of fish and stuff like that, and there's large fish die-offs because of it. And then if you eat those fish that die, then you would also get the BMAA. But most people don't eat dead fish that they find on the beach. Anyway, so here's serine, here's BMMA. Yeah, here's the red tide thing I was just talking about. You guys can stop me anytime if you got questions, you know. What about autoimmune diseases? They may or may not be metabolic diseases. And of course, the treatment, unfortunately, would be immunosuppression. So there's at least 80 autoimmune diseases. And autoimmune inflammatory diseases are basically the same thing. Autoimmunity, the patient's immune system is activated against the body's own epitopes, also known as an antigen. And in inflammatory diseases, the overaction of the immune system and a subsequent downstream signaling causes problems. Okay? I wish we could go into more information about the immune system to help you understand that, but we just don't have time. So, antiphospholipid syndrome. Okay, what the heck is this? So you get antibodies produced against membrane phospholipids that came from your own body. That will coagulate these things and can cause blood clots in various locations. So sometimes antibodies can attack prothrombin, turn it into thrombin. That can be a problem. Let's see. Treatment basically involves anticoagulants. So you find antibodies against cardiolipin, and you find antibodies against apolipoprotein H, and that's what they use in blood tests. Some of the people that start off with antiphospholipid syndrome seem to end up with lupus. So lupus is called systematic lupus erythematosus, seems to be related to antiphospholipid syndrome. So you have antibodies that attack ribonucleoproteins and other kinds of problems. And that leads to apoptosis of cells that shouldn't be dying. You can get inefficient apoptosis, and that can also lead to what's called necrosis. So to make sure you know the difference between apoptosis and necrosis, apoptosis is programmed cell death. So for example, at some point when you're an embryo in your mother, you're a fetus, you're actually, you have webbed fingers and toes. But at some point, that webbing, those cells are programmed to die so you get your individual fingers. That was meant to happen. When you injure yourself, say you get a cut, and that cut heals nicely, what that means is that the injured cells that said, okay, I'm going to die, I'm out of here, and new cells are replacing them. But when you get a cut that leads to a scar, or you have an infection that leads to a scar, or you get bit by a snake or something really toxic like that, and that can lead to necrosis, random cell death, and the remaining cells don't quite know how to handle that. So I have a friend who has lupus. I can tell you that she is extremely cautious about going anywhere with this COVID virus out there. So it affects all kinds of different organs. There are certain things that are characteristic, like the rash on the face, photosensitivity of the eyes, arthritis, things like that. And basically what they're saying is that apoptosis leads to, instead, necrosis. And these cells are necrotic, they're dying, they're wounded, they're giving off all kinds of cytokine type signals and things like that attracts cells that are going to attack it. And then you get other cells, white blood cells that normally engulf to breathe, they're not able to do it because they've been attacked by other immune cells. And yeah, it's just a problem. And yes, I put celiac disease in the section on autoimmune disease. So gluten produces a 33 amino acid peptide called gliadin. And that peptide is difficult for proteases to hydrolyze, giving more time for transaminases to start removing amino groups. And now it doesn't even look like a peptide. And so it causes all kinds of problems. Wait, where'd that one? It's not the diagram I thought I had in here. Anyway, so let's see, here's your resistant peptides before deamination. Here's transamination going on over here. And then they go into this antigen presenting cell. This is part of the immune system that starts to make antibodies in particular to it. And the next thing you know, you got antibodies that are attacking this gliadin protein. Okay. So we talked about it in more detail in chapter 29. But I just wanted to just reemphasize that, yes, this is really an autoimmune disease. And then back to diabetes. Could diabetes also be an autoimmune disease? Why do beta pancreatic cells get targeted for destruction? So a couple of years ago, so they created an antibody that would attack mature white blood B cells, and they were able to protect the beta pancreatic cells in mice because the B cells were attacking the beta pancreatic cells in mice. So this antibody drug prevented them from getting diabetes, and their blood glucose levels remained regulated. So at least in mice, it appears that some mature B cells attack beta pancreatic cells in an autoimmune mechanism. And so the incidence of type 1 diabetes has been increasing rapidly, and so has our exposure to viruses been increasing rapidly. So is there a correlation? And so the enteroviruses are found in intestines and in suids. They have a complex effect on mice. And anyway, there's no specific enterovirus that can be held responsible, but essentially they're taking mice and exposing them to enteroviruses from suids and other places and looking at the effect on diabetes. And there is an effect. So what they're finding is that old mice in general have a risk, and some younger mice also have a risk. Okay? So the importance of these experiments is that we're going to be able to look at, okay, what happens, what's the difference in risk between this type of mouse that's young and this kind of mouse that's young, and also what's difference in risk between the young mouse and any kind of older mouse. Why does age have anything to do with it? Okay. Well, we do know that T cells are activated by viral attack and all this kind of stuff, and it probably has something to do with what's called T regulatory lymphocytes. And so younger and older mice with these T regulatory lymphocytes fortify it against autoimmune T cells and don't get the disease. So that's what they're looking at now, is what are the effect of these T regulatory cells? Okay. So now they've looked at this, they figured out, well, what are we looking at and what's the difference between them? And it seems to be their T regulatory cells. They're calling them Tregs in some places. So chronic fatigue syndrome, I haven't worked on it now for several years, I don't have funding, but that was our premise. We just assumed that chronic fatigue was an autoimmune disease. Metabolic syndrome, also most likely an autoimmune disease. That's also closely related to chronic fatigue. So then I went into brain diseases or neurodegeneration. So Huntington's disease is an inherited and fatal brain disorder. Proteins that are produced in the nucleus clump together and cannot exit into the cytoplasm through the nuclear pores. So you get a traffic jam there and the cells die. Hepatitis viruses may be related to Parkinson's. They found that those with hepatitis B had a 76% greater incidence of Parkinson's. And hepatitis C, a 51% greater incidence. In contrast, those with HIV or other hepatitis, A, for example, had no increased risk. So then Alzheimer's. So we talked a little bit about Alzheimer's in chapter 29. 6th leading cause of death in Americans. It's growing. There's no cure, any proven methods to slow the progression. some major drug companies simply have given up. And because there's no treatment, insurance companies are using that as an excuse to not pay for the diagnostic tests. Well, why should we pay for it if it's not going to do you any good? Well, death from all these other diseases have decreased, but Alzheimer's is increasing. Yeah. So what about the beta amyloid plaques? So we know that the beta amyloid plaques trigger conversion of tau protein into tau tangles. These then bind to the nerve dendrites, turning them into a tangled mess. But no one knows why that happens in some people, but not in others. But it's more likely that the plaques associated with the disease may be a risk factor, but not the actual cause of the disease. So there's strong evidence that beta amyloid buildup kills neurons and synapses. There's families that have early onset Alzheimer's. And about 25% of people in the world carry one copy of this gene, and that doubles or triples the risk for Alzheimer's. Having two copies of that gene makes it 12-fold increase. Mice that were engineered to overproduce beta amyloid tend to develop memory issues. But then drugs and therapies designed to attack beta amyloid are simply not effective. Beta amyloid is one of those proteins that are known as intrinsically disordered proteins. It doesn't have a specific shape. It changes all the time, depending on what environment it's in. And that's one reason why it's very difficult to attack with a drug. So there was, I want to say hundreds, but a lot of drugs in clinical trials so far. So there are some that may show you some effects about reducing some of the plaques. More recently, they found this enzyme. This is the enzyme that cuts the protein that makes the beta amyloid. Okay. So the enzyme cuts presenilin. So this is the name of the enzyme. And Alzheimer's mice with reduced enzyme activity saw a reduction in the size and quantity of plaques in their brains. But you can't completely inhibit that enzyme because you need it to do its normal function. So PET scans. The basic way to look at brain plaques is very expensive. Like I said, insurance companies don't want to spend the money on it. But there was a very interesting study done a year or two ago where they used a simple eye exam used to detect plaques in the brain. So they correlated it to PET scans, which are the gold standard, and there seemed to be an excellent correlation. The eye exam was to be able to detect plaques in all 17 of the patients that were detected using PET scans. Well, what about Alzheimer's and inflammation? Makes sense. Inflammation, which could be caused by excess sugar, leads to malfunction of proteins because of glycation. Another thing that they're looking at now is sleep deprivation. So the increase in diabetes parallels the increase in Alzheimer's. There's a whole bunch of different correlations over here. Sleep-deprived mice had twice as much tau protein. So yeah, it makes sense to look at this stuff. It makes sense to regulate glucose levels by exercise, all these different things. People with type 2 diabetes more than twice as likely to get Alzheimer's. There's a lot of correlations. So correlation does not apply cause and effect. So, but some patients have seen large improvements by increasing their sleep, reducing carbohydrates, fasting. In other words, they don't eat all night. They eat their dinner and then that's it. They don't eat until the next day. Some supplements and exercise. So there's ongoing research in the area, but probably most of it is popularized in this book right here, the Bredesen's Protocol. So if you're interested in that, you can go there. But you know, they're making money off of it. We have a relative in Waimanalo that's developed Alzheimer's quite quickly, especially after his wife died and he became very depressed and didn't exercise and everything. And we tried to get him to exercise and take care of himself, but he just wouldn't do it. I think he was just too depressed, you know, because of the loss of his wife. So there was a study done from 2009 to 2011 in Finland. And so half of the control group were given regular health advice. The other half also regular health advice. But they were put on a Mediterranean-style diet, given strength and aerobic training, given cognitive training, and then they kept assessing them. And the lifestyle change getting significant improvements in every category. Significant improvements. Even those that had the gene variant associated with higher risk for Alzheimer's seemed to get greater gains. Okay. So here's some of the metrics they were using right there.
What's the relationship between dementia and Alzheimer's?
Well, basically, dementia is just simply loss of, mental function, right? Is measured by tests. And Alzheimer's is a very progressing disease. You can watch it progress, and the way you see it is with, the beta amyloid plaques. So with dementia, you may not have amyloid plaques. It may be a different kind of dementia. So for example, say you've had injury to the brain from physical injury or injury due to stroke or something like that, that would be a dementia.
Okay, thank you.
So this is something new. I just threw it in here. I find it extremely interesting. Immunity in the brain. So until recently, and I'm talking less than 10 years, maybe 5 years, they started doing this. The brain and the immune system were considered to be separate systems that didn't even interact because of the blood-brain barrier. They knew that the microglial cells in the brain were behaving in kind of an immune cell fashion to protect the nerve cells. Okay? But there was no evidence that peripheral immune cells, such as T and B cells, which is part of adaptive immunity, could enter. But they did know that there was immune cells found in the central nervous system during certain diseases. And then later they discovered a protein called integrin that allowed immune cells to cross that barrier. That protein is now used to treat some types of multiple sclerosis. And then they found that in a mouse model for Alzheimer's and ALS, when B cells and T cells were suppressed. That's most of your adaptive immunity, right? Not your innate immunity. The diseases progressed rapidly. And when you restored adaptive immunity, the diseases slowed again. So here's brain, central nervous system diseases that are obviously affected by the immune system. Mice whose adaptive immunity was suppressed experienced a several-fold increase in the severity of PTSD. They also had reduced spatial learning and reduced social interactions. And then even more recently, they discovered that the meninges, which was thought to simply help reinforce the brain, keep the brain separate from other things, does much more than simply reinforce the blood-brain barrier. Meninges contains peripheral immune cells that communicate with the brain by producing cytokines. Those are simply signaling proteins, right? So they found that the cerebral spinal fluid from the meninges enters the brain through the blood vessels and carries these cytokines into the brain via this newly discovered, what they call the glymphatic system. I don't even know if that name is going to stick, it's so new. And they able to influence neuron behavior. So one example is a cytokine called interleukin-1 beta that seems to initiate the behavior of sickness. You ever notice how people can start acting sick? Like, you okay? Yeah. Why are you acting sick? Sleeping, loss of appetite, reduced social interaction, right? Obviously, this is in mice. And then another discovery is that the presence of the lymphatic system in the brain. So lymph normally carries waste away from tissues, and including antigens, things that could cause an immune response. So they found that these antigens and lymphatic systems are presented through the immune system at the lymph nodes. And you know, probably from your own experience, that your lymph nodes get swollen. That's like you're fighting something, right? That's the reason why. So it was found that brain injury provoked a strong immune response in the lymph nodes outside the brain. So now they know that there's a way for lymph to exit the brain and interact with peripheral immune system. So why would the brain not have a regular immune system, both innate and secondary? Okay. Probably it's because of inflammation. So inflammation is kind of like a crude weapon. It causes damage. It kills good guys as well as bad guys. And the central nervous system is a place where cells don't regenerate. It's the most specialized part of your body is the brain. So you don't want inflammation happening in the brain. And that leads into another part where normal repair mechanisms go wrong. So inflammation. So acute, maybe short-term inflammation is helpful because it helps fight infection, heal injury, help rid the body of waste. And you have these cells called sentinel cells that alert your immune system, causing capillaries to leak plasma in order to envelop and slow down a virus or a bacteria. And then the macrophages release cytokines that activate T and B lymphocytes that then attack the antigen. But chronic or long-term inflammation is bad because it also activates the immune system. But there's no specific antigen to attack. So the system might start attacking itself in an autoimmune type response, causing all kinds of problems. So chronic inflammation can also cause even dementia, we were talking about, right? So obesity, stress hormones, lack of exercise and sleep cause chronic inflammation. Belly fat causes release of cytokines, and poor diet. And maybe that's why belly fat is associated with risk of certain diseases, right? Including cancer and heart disease. So very recently, and I don't know why I don't have a year on there, probably 2 years ago, they found that when monocytes are stressed, they shed parts of their membranes as what's called microvesicles. And these transmit alarm signals to other cells. So some of these microvesicles contain stress mitochondria, and they believe that these stress mitochondria release all kinds of reactive oxygen species that will then trigger inflammation. So anyway, they found associated with this tumor necrosis factor, and they also found interferon associated with these microvesicles. So people are looking at how do we either stimulate the immune system to get rid of the microvesicles, or how do we prevent them from forming? And ferroptosis is a newly discovered form of programmed cell death. It's distinct from apoptosis. It's meant to kill damaged or malfunctioning cells. So normally, lipid peroxides are destroyed by the glutathione reductase pathway. But this protective pathway has simply been overcome. So it's these, like here's an unsaturated lipid, you initiate this on purpose to produce radicals, and then go and attack the cell. So these are all iron-dependent processes. That's why it's called ferroptosis. Like apoptosis, ferroptosis. So then bacterial and viral infections. In fact, drug-resistant tuberculosis has surpassed AIDS as the leading cause of death by infectious disease. They were able to kill this one strain by taking 3 drugs at a time. And the only vaccine available is mostly used to protect babies, since it is not very effective once TB is in the lungs, because it just basically hides in the lungs, coats itself, and you can't get to it. And recently, a brand new article came out like about a week ago. I sent it to some people that were involved in vaccines for the COVID-19. It correlated that people who were given this vaccine against tuberculosis had a much less infection and death rate. A really interesting correlation. So here was a vaccine made to a bacteria, and it seems to be helping protect people against the COVID-19 virus. So bacterial collections have been held since 1915. And so why do we do that? Because essentially, the more you know about a potential enemy, the more you might be able to defeat that enemy. So they use these collections to study bacterial evolution and a bunch of other things, right? So let's see. Going back to the tuberculosis. And so they studied tuberculosis that happened in, I guess, 1999 or 2000 something. There was a big, I forget, I didn't add it. But anyway, so there was a big outbreak. I think it was 1999 or 2000 something. Anyway, so they took that strain of TB and they were able to trace back to 3 mutations that happened in that organism between the early 1980s and 1993 that resulted in that present organism. So with that information, they can then sample tuberculosis bacteria in different parts of the world and see what state they are at. Are they potentially going to turn into a more virulent form? We can isolate them, grow them, and find out what we use to kill this particular variation. And this is all in a greater umbrella of bioinformatics. Because of computerization and everything else, we can keep track of all this stuff now. So here's an example of a human disease network. If you blow this up, you can see that the size is related to the size of the outbreak and where the outbreak went, how they're connected. So it's like contact tracing for every disease in the human population. Okay, let's see. So that's it. I'm glad we finished that chapter today. So any questions about anything in this chapter? So anyway, I mean, the purpose of me putting this chapter together was not to make you memorize a bunch of stuff. Okay, you have enough stuff in the basic understanding that you need to know. But for one, it's examples of all the different things you've learned. And hopefully it will spur your interest and help you write your extra credit papers. And maybe, who knows, you'll become a researcher and you'll cure a disease. That's what got me into science. I thought I was going to help cure cancer. I did get to work at the Cancer Center for a couple of years, but then really didn't turn out much. So no questions? Okay, well, we have one more chapter to go. I'm still kind of editing it. I'm not real happy with it. It's too big and unorganized, but that's the problem. And then we'll have time for a review before the final. And then the final can be taken on Friday or it can be taken on Monday. So if we don't have any more questions, I'll just sign off and then I will see you folks tomorrow, most likely at 1:30, not noon.
Thank you.
Have a great day then.
Thank you.
Take care.